Hello, everybody, and welcome to AMP's Digital Experience. I'm Dr. Julia Gouffon, a Translational Applications Liaison with Mission Bio, and we're proud to be sponsors of this series. And today, I just wanted to start speaking to you about unleashing the power of single-cell multimodal analysis to advance precision medicine. And then I'll pass it on to Dr. John Lee to give his presentation. So Mission Bio is a leader in single-cell multi-omics. We started back in 2014, spun out of UCSF, and now we've grown into well over 100 employees and launched our Tapestry instrument in December of 2017. We're in over 50% of the NCI cancer centers. We have worldwide users, and our expertise is really in designing DNA panels to look at SNPs, indels, and also known rearrangements. And then starting last December, we also launched protein panels that you can look at cell surface proteins and understand, which is the cell population from which these DNA mutations are occurring. We work with many leading institutions, research institutions around the globe, and also have access to some CLIA labs that can run samples for you. And then we also have internally a pharma applications development laboratory to work with pharmaceutical companies that are interested in developing their single cell technologies. And the reason we exist is that we believe that in order to truly understand cancer evolution and be able to knock out cancer in the body, that we need to understand cancer disease at a single cell level. We know that we start out with a first hit that grows to produce cancer, and then normally one might do some kind of bulk sequencing. And the problem with that is that you get variant allele frequencies for all of this entire population, and that confounds the data. So you don't understand that perhaps these cells are germline cells. They don't have any kind of mutations in them. And here's a very rare cell population that's present, and then it might become the driver of clonal resistance. So we feel that we need to really look at each of these cell populations on an individual level to understand the co-occurrence of these mutations and to detect these very rare cells. So again, as you can see, the limiting analysis of bulk NGS is that we're looking at a population average, which is ambiguous or can even be inaccurate. You can't detect the germline versus the somatic cells very well, whereas we can do that. And we're also able to look at the zygosity. Do we have heterozygous, homozygous, or mutations? We can look at the co-occurrence as well. This cell only has one mutation, this has another mutation, but this cell population has both of those mutations occurring at the same time. And we can do a 0.1% on SNPs, whereas usually with bulk sequencing, you're somewhere around 2 to 5% variant allele cutoff in frequency. And then when you're sequencing, often you're just measuring a single analyte, whereas we can do multimodal analysis now, where we can do SNVs, CNVs, also look at those cell surface proteins, and then fully resolve all of these in a multimodal analysis to look at all the different cell populations. And this is the instrument that we do this on. It is a single sample workflow, but multimodal analysis to where you can get your DNA and your protein simultaneously. How does it work? We're really a system or a platform. So we first have on the left-hand side here, Tapestry Designer, which is what we utilize to enable the customer to design their own DNA panels. We have the help of FAS, or we can even do a white glove service with our own bioinformatics group. As far as looking at cell surface proteins, we do work with BioLegend. We have a Total-seq-D panel, which covers pretty much the entire hematopoietic differentiation cascade. We can also do spike in with custom cell surface proteins if you have those and want to have them conjugated by BioLegend. Here's our instrument. It sits up front of many of the different Illumina sequencers. It depends on the size of your panel and how many samples you're running and the number of cells that you capture, which sequencer you would utilize. But the data comes out as a FASTQ file for both your protein and your DNA libraries, and those can be uploaded into the Tapestry pipeline. The Tapestry pipeline is cloud-based, and it is utilized to do the alignments and filtering of the data, and it is also made available free to our users. And then we have the data that comes out of here. The Loom files goes into Tapestry Insights, so you can look at longitudinal data in these fish plots, or you can look at clonal phylogeny, and many other types of insights can be provided. How does it work? We differ from other groups in that we have a two-stage workflow. The first one, the cells are flowed and encapsulated into single cells and individual oil droplets with a lysis buffer and protease K. And what that does is when we heat activate it, it removes those histones and other proteins that are keeping the chromatin bound in a tertiary structure. So that linearizes the DNA, and that gives us access to provide in the second droplet step unique molecular identifying barcodes, PCR primers and reagents, amplify those areas of interest, and then we reduce sequencing errors and copy number errors by only amplifying about 9 or 10 times. So we start out with about 100,000 cells here. We flow them on the system. We capture about 5 to 10% of them. So you're really looking at a population of, say, 5 to 10,000 cells that come out on the end. Now, if you're interested in doing the antibody oligo staining, you can do that up front here. That's done offline, but these oligos are specially, antibodies are specially tagged with our antibody, and it has our capture sequence on it so that we can amplify those during this particular step. After they are sequenced on the NGS platform, what we end up with is simplistically here an IDH1, IDH2 mutation. We know the exact mutations that we have. This cell population has an IDH1, NPM1, and a TP53 mutation. And then we know which cell population contains these mutations. So to date, we have many panels that we have designed due to starting out in the heme field. As you can see here on the left-hand side, these are our catalog panels that are ready to ship at any time. And then recently we have several published panels that are from research groups that have published and are allowing access to their panel designs. And then we have these virtual panels that are in silico designs that you can also utilize and use as a jumping off point. For the solid tumor, we have a tumor hotspot panel that you can utilize, but most of our solid tumor groups are designing their own panels, and we're working with them right now to perhaps release some of the published data that we have and utilize those panels coming up soon within breast cancer, GBMs, and melanoma, for example. And then one of the largest areas of growth that we have is looking at cell and gene therapies, where people are doing CRISPR edits and really need to understand what is happening off target and on target. This is really the most accurate way to do that, to make sure that you don't have any rare occurrences happening that you cannot detect by simply looking at digital PCR. We're able to upload any kind of reference, so we can do a human, mouse, or any other custom reference genome that you happen to have as well. And this is, as I said before, the Total-seq-D antibody oligonucleotide. We have a menu of over 150 individual Total-seq-D antibodies that have been conjugated now. So that could include PD-1, PD-L1, CTLA-4, for example, for solid. But you can see this is how we're able to look at all of these different monocytes and memory cells, T cells, B cells, and understand what's happening in each of those from a genotypic level. And due to customer asks, we have now started to where a lot of people said we can't get 100,000 cells can you lower the input? We are able to load about 20,000 cells and then still get the exact kind of data, panel uniformity, and output dependent upon even if you do load a lower amount. So this will help us in conserving precious samples. We're also working with groups to try to develop ways to isolate circulating tumor cells, for example, and be able to put those on the cartridge and get genotype data out of those. So the type of samples that are amenable to the platform at this point, any kind of fresh frozen, even non-viable samples can be run on the platform because we're looking at DNA only. So we are able to flow nuclei on it. Cryopreserved samples in DMSO, PBMSCs. You can fax sort and enrich for certain populations up front if you'd like to. We can, of course, do dissociated fresh frozen tissues. We work with PDX models and cell lines. So I just wanted to give you an example here of the value of looking at tapestry in therapeutic responsive relapse. This is a paper done by Alexander Pearl, Kathy Smith, and McMahon and Cancer Research, looking at patients that have AML and were given gilteritinib. As you can see in this particular instance here, on the left-hand side, you have a varying allele frequency around 20%. And at baseline, they thought that this patient's clonal population had a FLT3 ITD co-mutated along with an IDH2 and an SF3B1 in the same populations. However, at the end point, the FLT3 ITD, which is the target of gilteritinib, decreases, whereas we continue to see an upward trajectory for IDH2 and SF3B1. So they decided to run this on the Tapestry instrument. And what they have here is you can see this clonal population that had an IDH2, SF3B1, and the FLT3 ITD in it did decrease in response to therapy. However, we have this IDH2, SF3B1 clonal population that was small at the outset, and that continued to grow over treatment as it was not targeted by the drug. Since we have IDH2 inhibitors now, it's possible we could have done a combination therapy at the outset and knocked out these clones. We also have a quadruple mutant NRAS clone that developed, and they wondered at what point they could detect the NRAS. They went back and pulled a couple of on-treatment blood samples that they had, or bone marrow samples that they had. They were able to detect 3 cells out of a total of 7,725 cells, and that equates to 0.04% of the cell population. Because we have data points at 4 different loci for each of these amplicons, we're able to definitively say that we could detect those cells. Another area that we are being utilized in is in hematological measurable residual disease. And there's several ways that one can look at that. One would be doing NGS, of course, one would be doing flow cytometry, but we feel that in combining the cell surface proteins along with the ability to look at the genotype, we can not only detect it, but we could say which cellular compartment is it present in, and is that going to be important in the treatment. So this is data out of Stanford from Dr. Ravi Majeti's laboratory, and it shows just simply 3 different ways that CHIP can occur, not CHIP, but MRD can occur in patients. One here is that the relapse clone was the same one present at diagnosis, and we can detect 6 cells out of about 5,000 cells at remission for this patient. Here we have where the relapse clone and the diagnosis clone are similar, but you have this confounding data, which are these CHIP mutations occurring in the background. So often this confounds data, we're not sure what to do with it, but combining the immunophenotype along with the genotype, we're able to resolve that. And lastly, we have a clonal remodeling where we can't detect the clones present at relapse, but we could detect them during remission. We have 10 cells here out of a total of 4,000 cells. So this is something that we're working with several groups to develop, a clinical assay that might be developed into a lab-developed test at these facilities. And this is just showing some data to show that we have CHIP occurring, which is we have the SNPs occurring here that are similar for these two cancer populations. We have C and Vs occurring, and you can see we've got copy number loss in each of these areas. And then we have cell surface proteins. It's only in using these cell surface proteins that we can see that there's two separate cancer populations occurring here. And then we still pick up the T cells and the B cells. And this is in some solid tumor data. Here we have patients that were done on a rapid autopsy sample, and they took the lung metastases, the chest wall metastases, and the liver metastases and ran them on a panel. And you can see that there's two different trajectories occurring. On the left-hand side, we have an NRAS and a BRAF clone population that becomes 70% of the lung metastases, whereas on the right-hand side, we have NRAS drivers of the chest wall metastases and the liver metastases. And lastly, as I said, we have several people looking at this for gene therapy, looking at cell and gene therapies, where we need to make sure that we do the proper edits. And they were trying to affect homozygous edits at 3 different loci. When they did the bulk sequencing, they thought that they had this at about at least 11% occurrence. However, when we ran it on single cell, you can see that it's only 2.91% for each of those homozygous edits to occur. They went back and did design iterations and condition optimizations, and they were able to get that up to 52%. So when it is important for these cell and gene therapies that are going to go into patients, we have to make sure that we have the correct edits being done. So this is a list of our publications that we have right now. You can certainly find out more information by going to missionbio.com. And with that, I would like to turn it over to Dr. John Lee from Fred Hutch, and then we can take questions at the end. Thank you.
Great. Thank you very much to Mission Bio, as well as to AMP for allowing me to share some of the work that we've been doing in the laboratory. And I'll talk to you today a little bit about some of the combinatorial genetics work that we're doing to look at genotype-phenotype relationships in bladder cancer as well as prostate cancer. So as a disclosure, I am on the Speakers Bureau for Mission Bio. And so let's start with bladder cancer. And bladder cancer is really marked by heterogeneity at a number of levels. And one of the initial characteristics that we noted about bladder cancer is its relatively high tumor mutational burden. And when you take a look at one of these oncoprints, specifically the TCGA muscle-invasive bladder cancer data set, what you rapidly identify is that here, shown in rows, are the specific genes that are altered at high frequency in the disease. And each of these hash marks in the columns represents an individual tumor. And so it's pretty evident that there are a number of mutations that can occur in a single tumor. But when you compare one tumor to the next, the spectrum of mutations that are present are quite different. And if you tabulate this, we find that there are at least 30 genetic alterations that occur in muscle-invasive bladder cancer at a frequency of at least 5%. And on average, each tumor has 5 genetic alterations just within this set of 30 or so genes. Beyond the tumor mutational heterogeneity is the phenotypic complexity of the disease. And so there is this molecular classification. It's a consensus molecular classification that's been devised. And so at the histologic level, while these tumors may look like urothelial carcinoma, there's clearly transcriptional differences that define them. And so they can be broken up to basal squamous, various luminal subtypes, the stroma-rich, and neuroendocrine subtype. In addition to the more common urothelial carcinoma histology, there are also histologic variants that are often intermixed into these tumors, and these can include sarcomatoid carcinoma, macropapillary carcinoma, small cell carcinoma, and plasmacytoid carcinoma. And so to take a step back and think about that degree of complexity that we see at the clinical level versus the types of models that have been able to be generated in the laboratory, what we know is that these genetically engineered mouse models of cancer are really the gold standard. And so to give you a brief history of 30 years of gen modeling in bladder cancer, the next 3 slides will go over a variety of these transgenic models. So initially there were UP2 specific promoters driving various oncogenes. And essentially, these showed pretty mild phenotypes, in most cases, precancerous lesions or hyperplasia. And then there were additional Cree-based drivers to recombine various potential tumor suppressor genes in the context of transgenic expression of oncogenes. And again, here the phenotypes are relatively mild, in most cases, relatively non-invasive disease. And only recently, with the use of the delivery of adenocrete to the bladder in the context of flox alleles, specifically, for instance, P53 and P10, have we been able to see really advanced muscle invasive aggressive bladder cancers that do metastasize. And so one question is, why is it so difficult to model the spectrum of bladder cancers, specifically aggressive bladder cancers, in GEMS? And so one hint as to why this may be the case came in a set of back-to-back papers that was published in Science in 2020, where they looked at histologically normal urothelium from both transplant donors and looked at the mutational spectrum and compared it to the mutational spectrum of bladder cancers from patients. And surprisingly, what they identified was that there were putative oncogenic mutations that accumulated in histologically normal urothelium. And what this suggested was that potentially the urothelium could be tolerant of multiple mutations before the onset of malignant transformation. And so we asked the question, how can we more efficiently model the complexity of muscle-invasive bladder cancer in a genetically defined manner? And this question would then lead to the next question of how does genotype relate to phenotype? And certainly we had to acknowledge some limitations in terms of available technologies. For instance, with the genetically engineered mouse models, relatively few genes and gene combinations can be modulated or manipulated in that setting. And that may certainly be a reason why the types of bladder cancers that have been modeled have underrepresented the phenotypic heterogeneity of human bladder cancer. And so we wanted to use more modern technologies, for instance, organotypic or lunoid cultures, which allow for scalable and facile ex vivo manipulation. And then to also incorporate single cell sequencing, which might allow for massively parallel analysis. So this is a basic model of what we wanted to achieve. And so we wanted to generate a lentiviral library of genetic perturbations associated with bladder cancer, and then to introduce these into target cells, and target cells being primary urothelial cells. And if we could introduce this lentiviral library at a high multiplicity of infection, such that each of these primary urothelial cells receives a random combination of genetic perturbations, then perhaps a subset of these cells would have received sufficient signaling to then become transformed. And then we could use targeted single cell analysis to identify the specific genes that were involved in the development of cancer. And hence we could associate compound genetic perturbations with complex phenotypes. And so the first question was, what cell population do we start with? So clearly the urothelium is pictured here, and there is a urothelial hierarchy, and there are umbrella cells that face the bladder lumen. These intermediate cells, and then basal cells that line the basement membrane. And so one characteristic of the basal cells, which I'll talk about more here, is the expression of sonic hedgehog. And work from Phil Beachy's lab really has defined that sonic hedgehog positive basal cells are the cell of origin of muscle-invasive bladder cancer. And this was established through a set of elegant experiments wherein his group use these sonic hedgehog Cree-ER mice with a ROSA 26 MTMG allele. And basically these mice were treated with tamoximet in order to convert the tomato to MGFP in this sonic hedgehog positive basal cells. And then these mice were treated with BBN, which is a urothelial carcinogen, for 6 months to initiate invasive carcinoma. And what they found at the end of that was that indeed they were able to generate these invasive urothelial carcinomas, and that these invasive urothelial carcinomas were marked by GFP expression, indicating that they had been initiated from sonic hedgehog positive basal cells. They went on to perform another elegant experiment in which then they determined whether the sonic hedgehog positive basal cells were really necessary for the initiation of a urothelial carcinoma in this setting. And here, what they used was this ROSA26 allele driving diphtheria toxin. And so in the context of tamoxifen, you then activate diphtheria toxin in a sonic hedgehog basal cell specific manner, thereby eradicating the basal cells. And so in the absence of tamoxifen, if you treat with BVN, you get these tumors that grow out as anticipated and shown previously. But when you treat with tamoxifen and ablate these sonic hedgehog positive basal cells, then treat with BVN, you actually do not form tumors. And so this really established that the sonic hedgehog positive basal cells are really the cell of origin, and that ablating these basal cells confers resistance to carcinogen-induced bladder cancer. And so around the time that we were starting this work, a group from Spain had started to define different immunophenotypic markers of basal and luminal cells in the bladder. And so they had identified that EpCAM and CD49F was able to mark a population of basal urothelial cells that could initiate organoids at a very high frequency. Whereas the cells that were CD49F low, or the more basal cell-like, could not initiate organoids very efficiently. And so we took this knowledge, and one of the gaps that we had to overcome was the low efficiency of lentiviral transduction of primary cells. And to overcome this, we basically isolated these CD49F high, EpCAM positive mouse bladder urothelial cells, and we put them into Measurable as cell suspensions. And into these cell suspensions, we actually admixed concentrated lentivirus. So in this experiment, we use concentrated GFP expressing lentivirus. And so we would mix the virus, the cells in the Measurable, and allow them to solidify after plating. And what this allowed is for an enforcement of contact between lentivirus and the cells. And what this allowed was a significant improvement in the efficiency of transduction. And what we can see here is that with as little as 5 microliters of GFP lentivirus per 15 microliter organoid droplet containing 20,000 cells, that you could achieve 100% transduction, and that this could be titrated. And so with decreasing doses of lentivirus, you could have sort of a linear relationship between dose and percentage transduction. And then importantly, when we looked at GFP copy numbers, it became apparent that we could achieve up to 20 copies of lentivirus per cell. So this really enabled us to achieve a high MOI transduction within the system. And so being able to achieve high MOI transduction, what we wanted to know is whether we would be able to really enumerate the specific lentiviruses that had integrated into these cells. And so we conducted this experiment where we generated barcoded lentiviruses that express GFP. They're barcoded both at the 5' end and 3' end with 10 nucleotide barcodes. We worked with Mission Bio to create a custom single-cell DNA amplicon sequencing panel. And so we had amplicons that involved primers to amplify the 5' barcode, 3' barcode, GFP, and then a number of endogenous mouse genes as controls. And so we moved on to then generate a cell population, 3T3 cell population, some of which were unlabeled, some of which were labeled with 4 different barcodes, or 3 different barcodes, or 2 different barcodes. And we mixed these cells together so that we knew the expected frequency of these cells. And then we took this pool of cells and ran them on the tapestry using this amplicon sequencing panel. And what we found was that the tapestry really did a great job of identifying each of these populations. And you can see that by comparing the clonal frequency identified versus the expected frequency, that they match up fairly well. But most impressively, it was able to identify this one population with 2 barcodes, barcode 5 and 7, at an which was present at the expected frequency of 0.14%, and it was identified here at 0.52%. So we were confident that this would be a sensitive approach and panel that would allow us to move forward with our experiments. Another question that came up was in the context of this lentiviral transduction in matrigel and organoids, whether the distribution of lentiviral transduction would be normal. And so to evaluate this, we took our basal bladder urothelial cells, introduced a barcoded lentiviral pool at a multiplicity of infection of 2. After a couple days in orbital cultures, dissociated these cells and ran the single cell DNA amplicon, library preparation, next generation sequencing, and then plotted cell fraction by viral copy number. And we can see here is that this does indeed follow a binomial distribution, which appears to be normal. And so that was reassuring to us that there wasn't some sort of artifact where a subset of cells were highly transduced, whereas only for instance, the remainder of cells would not be transduced. So having established those technical aspects, we went ahead to create the library. And so we selected a variety of genes divided by gain of function or loss of function. And these were selected based on their identified frequencies in bladder cancer. So we generated 2 different lentiviral constructs, again, individually barcoded at the 5' and 3' ends, expressing either an open reading frame for gain of function events, or expressing a short hairpin RNA to recapitulate loss of function events. And so these short hairpin RNAs were selected from the RNAi consortium. They were tested by qPCR, and we selected those that demonstrated at least 60 to 70% knockdown. Another technical optimization that we had to perform really related to generating these lentiviruses in arrayed format. And so we had to generate each of these in arrayed format to prevent barcode swapping. And when we did this, we would then collect the viral supernatant, pool them, concentrate by ultracentrifugation, transduce our urothelial cells in organoid culture, isolate the genomic DNA, amplify the lentiviral barcodes, and generate libraries by 2-step PCR, and perform bulk sequencing to then look at the representation of each of the lentiviruses in the pool. And not surprisingly, we initially found that the shRNA lentiviruses were produced at much greater efficiency than the open reading frame lentiviruses. However, this information allowed us to then go back and adjust the producer cell surface areas in production of the lentiviruses and to go back around the circle. And when we did that, we were able to nearly normalize the open reading frame to shRNA ratios within our lentiviral pool. So having established a number of technical parameters there, this is really the experimental schema that we were left with. And so I've already explained many aspects of this, but essentially we take the primary basal bladder urothelial cells. We can choose to transduce these cells at a targeted mean viral copy number with this barcoded lentiviral pool. And once these cells have been transduced, we allow them to recover a couple days in the organoid setting. Then we dissociate these, combine them with the inductive embryonic bladder mesenchyme. And then in initial experiments, we've been performing subcutaneous grafting in immune deficient mice with the hope that a subset of these cells would then form tumors, that these tumors may or may not be oligoclonal or polyclonal, and that we could then perform single cell DNA amplicon sequencing to resolve the clonal architecture. If there was evidence of distinct histologic regions, that we could then perform laser capture microdissection and bulk DNA amplicon sequencing as well. So, the question is whether this works, and in fact, it does seem to work pretty well. This is one vignette, so this is one tumor of many that we've gotten out of the system. And so when you section this tumor and look at it after H&E staining, you can actually identify 3 clinically relevant histologies here. And so this region of the tumor actually shows urothelial carcinoma with squamous differentiation. This region here actually shows papillary carcinoma with inverted growth. And this region here represents sarcomatoid carcinoma. And so one of the first questions that we asked was whether these represent 3 clonal cancer outgrowths, or whether these might just be divergent differentiation from a common precursor. And so given that we have the single cell DNA amplicon panel from the tapestry, we went ahead and dissociated this tumor to single cells and performed barcode enumeration to identify whether there were specific clones that might be present. And so we identified 3 dominant clones from this analysis. This one clone was marked by this FGFR3 activating mutation, and this was of particular interest to us because FGFR3 mutations are enriched in luminal papillary urothelial carcinoma. As I'd shown you, there was one region of the tumor that demonstrated papillary urothelial carcinoma with inverted growth. The other thing that I would note here is that there are a shared set of common alterations between all three of these clones. And then again, clone 2 and clone 3 kind of have different alterations associated with them as well. So one of the problems with dissociating the tumor to single cells is that we lose all information about histology. And so it was very difficult then for us to go back and say, okay, this clonal population relates to this histology. And so to really confirm this, what we did was we took additional sections of the tumor and performed laser captured microdissection from those 3 distinct regions, isolated genomic DNA, and performed 2-step PCR, as well as next generation sequencing. And once we'd done that and matched up the barcodes that were present within the data from the tapestry analysis, we were then able to assign, yes, indeed, that this papillary carcinoma histology related to this clone that did indeed have this activating FGFR3 mutation. This red clone corresponded with the urothelial carcinoma with squamous differentiation, this green clone with sarcomatoid carcinoma. And so this system allows us to then go back and validate these findings. So what we can do is now take the mouse bladder, dissociate, identify, and isolate the primary basal urothelial cells. And rather than using that large pool, what we've done here is we've used the activating FGFR3 mutation as well as some of the alterations that were commonly found between those 3 clones that I'd shown you previously. And then performed a very similar transformation experiment and transplantation experiment. And what we found after the tumor grew out after several months was, in fact, the tumors demonstrated a luminal papillary carcinoma phenotype with inverted growth. So this really suggests that mutant FGFR3 is indeed likely a lineage driver of luminal papillary urothelial carcinoma. So I did mention to you that we've had several other examples, and mixed histologies are generally the rule. And so you can tell from this image at low magnification that there are 2 apparently distinct regions of this tumor. And when you stain with pan-cytokeratin, you can clearly see that this region to the north stains quite well. The region to the south does not. When you zoom in, you can also identify different cytologic features between these different cancer types. And so this is the diversity of models that we've generated thus far. And in stark contrast to what I showed you previously with the genetically engineered mouse models, we've actually been able to generate quite a few high-grade urothelial carcinomas as well as sort of low-grade papillary urothelial carcinomas. But quite interestingly, we've generated quite a few variant histologies as well that either are pure or intermixed. And these include sarcomatoid carcinoma as well as squamous carcinoma, and even some hints of perhaps neuroendocrine carcinoma, which we're still investigating at this point. So I wanted to switch now to some of the work that we're doing in a parallel system in prostate cancer. And so very similar to this mouse urothelial transformation system, there's also a parallel mouse prostate transformation system, wherein you can essentially isolate epithelial cells from the adult mouse prostate and mix them with embryonic urogenital sinus mesenchyme. And when you implant these under the kidney capsule or under the skin of mice, the inductive urogenital sinus mesenchyme can prime these adult epithelial cells to form prostate glands. And so in work done by Dr. Owen Whitty's lab over the last 10 to 15 years, they then utilized the system to show that you could introduce a number of oncogenes into the epithelial cells to understand how various oncogenic signals may cooperate to initiate prostate cancer. And in this example, they used human prostate epithelial cells to introduce AR, AKT, and ERG to then generate what appeared to be very similar histologically to human prostate cancer. So we've done something similar, but starting with mouse prostate epithelial cells, and we've isolated mouse prostate basal epithelial cells, again, using this EpCAM and CD49F high immunophenotype. And again, we showed that within this organoid transduction system, that with very little virus, that we can get near complete transduction and can achieve multiple copies of GFP per cell. So we did clone a barcoded library of lentiviruses that recapitulates prostate cancer-associated genetic events. You'll see that this is relatively quite different from the bladder cancer library. And one of the really interesting histologies that we've gotten out from our transformation experiments is this generation of a high-grade prostate cancer with pleomorphic giant cell carcinoma phenotype. So this pleomorphic giant cell carcinoma phenotype in primary prostate cancer is thought to be relatively rare, but is increasingly identified in advanced cancers. And generally what you see is that there's a very highly aggressive prostate adenocarcinoma component, and then there's this component where you see these very giant nuclei that can oftentimes be like 5 to 10 times larger than that of the adenocarcinoma component. And so we've seen this in a couple of tumors that we've generated this far, and to verify that these are in fact of a prostate origin, we have done some staining for androgen receptor. You can clearly see that these stain for androgen receptor. They also stain for HOXP13. And for the clinical pathologists, they often want to exclude the possibility that this has arisen from urothelial carcinoma, and so we've stained for GATA3, and these are negative, suggesting that this is not some sort of contaminated urothelial carcinoma. And so this provided us opportunity to try and investigate the sorts of genetic alterations that might be driving this phenotype. And so using flow cytometry as well as a variety of nuclear dyes, we were able to isolate from these tumor cells sort of small or large cells and to propagate these. And so what happens is that if you propagate the large cells, you have some population of large cells that persist, but then some of them revert back to the small cell population. When you take these smaller cells or normal cells and you propagate them, most of them remain sort of normal cells. We did perform these nuclear dye stains, and these are flow cytometry histogram plots indicating that indeed these larger cells do indeed take up more of these dyes. And so having established the ability to isolate these cells, we then went ahead and did tapestry analysis looking at the normal cell progeny versus the larger cell progeny. And what we found is that there was quite a diverse mix of genetic alterations with the normal cell progeny, but what really popped out in the larger cell progeny was that there was this inclusion of a short hairpin KMT2C or MLL3, and you can see that this is the level of enrichment that we identified within that population. And so, we're investigating now a potential link between MLL3 and pleomorphic giant cell carcinoma, and we think that there's probably a link through homologous repair deficiency and genomic instability, and I'll walk you through why we think this is the case. So there were two papers published in the last 2 years that have indicated that KMT2C mutation or loss actually leads to homologous repair deficient state. And this is thought to be due to downregulation of the expression of various enzymes associated with homologous recombination. And what this does is it then confers a state of genomic instability and sensitivity to PARP inhibition. And this follow-up paper that was published last year suggests that MLL3 may be even recruited to DNA damage sites. And here they also identified that the loss of MLL3 leads to enhanced PARP inhibitor sensitivity in cancer. So then what's the connection to pleomorphic giant cell? Well, it turns out that Jonathan Epstein at Hopkins had collected a series of primary pleomorphic giant cell carcinomas and performed, in conjunction with Colin Pritchard here at the University of Washington, some genomic studies. And what they found was that actually these focal pleomorphic giant cell carcinomas are highly enriched in various mutations in DNA repair, both in mismatch repair and in homologous recombination. And so we think that perhaps there may be a connection there that we are functionally validating currently. So in conclusion, I hope I've shown you that the technology offered by the Mission Bio Tapestry can be used in novel ways to potentially delineate genotype-phenotype relationships. Certainly, I would say that we're just getting started in this work, and all models are wrong, but some are useful. We're already thinking about ways to improve on this. We're using immune deficient hosts now, but perhaps we can switch over to immune competent syngeneic GFP tolerized hosts. We've been using subcutaneous implantations, but certainly we want to move into a more native microenvironment through orthotopy injections. And we have started to extend some of these studies to other tissue types, including those of the lung and stomach. And in terms of future applications of technologies that may be developed on the Mission Bio Tapestry platform, would be the incorporation of DNA and cell surface protein, or potentially the use of DNA and RNA in the future. And so with that, I would like to acknowledge members of my lab who've contributed to this work, the bladder cancer group, as well as the program in prostate cancer here at the Fred Hutch, folks at Mission Bio who have been really helpful and supportive through our conduct of this research, and my various funding sources. And I will stop there.
Great, thank you, Dr. Lee, very much. I have one question I've got to answer from the chat from my talk, which was, can we do FFPE samples? And at this juncture, that is not possible. We have tried some, tissue dissociation methodologies with the FFPE samples. If you can isolate nuclei, of course, you can flow those, but unfortunately, the cross-linking that occurs during FFPE makes it difficult to get those to flow. Then I do have a couple of questions, Dr. Lee. I love your talk as always. On the single cell, when you said you dissociated to single cell for the urothelial, papillary, and sarcoma laser captured microdissection. I know you didn't do that with those necessarily, but you did do some single cell. Did you flow nuclei or cells on our system at that point?
Those were cells.
Cells, okay. And then you kind of mentioned this as a moving forward, the ability, I was thinking, to use those immunophenotypic proteins on the cell surface to identify perhaps, are there distinct markers between the papillary forms or sarcoma and urothelials with the, that you could maybe identify the different cell types from cell surface?
Yeah, absolutely. I mean that's the idea. We're currently trying to identify such surface markers that we could use to potentially differentiate these phenotypes. And then the hope would be that, we would overcome that issue of the sort of like loss of histologic information with single cell dissociation that could be then recovered by using these surface markers.
Right. Okay, great. Yeah. I hope that works out well for you. And then I didn't know if you had thought about, we have another group that's developing organoid models and then doing, treatments, for example, with, as you said in the prostate there, a PARP inhibitor or something like that to see if they developed any kind of new mutations or resistance, if you would be able to utilize these organoids in that methodology.
Yeah, I mean, that's something that we've been thinking about as well, and we proposed it in a grant that's funded, and so we're going to be starting those experiments up pretty soon. In prostate cancer, there's quite a number of treatments. One of the major ones is androgen deprivation therapy, and so understanding genetic mediators of resistance to androgen deprivation, castration resistance, would really be interesting, but certainly it could be applied to a number of other active therapies.
Okay, great. Does the audience have any other questions that they'd like to initiate in the chat box there? We'll give them a couple of minutes to see how those are going. And so I guess this is, you're working with Matt, so he's our cell and gene therapy group. And do you have this in any kind of patient samples that you're also going to be utilizing this in to track their type of mutations for single cell? Or is it just
we haven't, we've just done it in the mouse models, and this was kind of like, we got in pretty early, obviously, because it was sort of a novel idea to use this for functional genomics and discovery. But I think this is certainly something that could be incorporated into a number of active clinical trials that we're involved in, and something that we've been discussing as well.
Yeah, okay, great. It looks very exciting. Yeah, I was looking at the KMT2, I'm sorry, I'm used to saying 2A because that's a therapeutic, I think that's in development right now, but I didn't know if there was one for KMT2C in development.
Yeah, I'm not too aware of anything that would be able to inhibit that.
Okay. Yeah, there's a couple of companies out there with menin inhibitors that are coming out, so or in trials right now. So okay, all right. Jocelyn says there are no other further questions. So with that, I guess we'll close. And I appreciate everybody listening in today. Please do go to Mission Bio. We're happy to speak to you about your experiments and see how we might be able to design a panel for you to look at any kind of DNA mutations that may be occurring in your system. And we appreciate Dr. Lee getting up early for us today to present. It was very, very useful, I hope, and thank you for your time. All right, thanks.